to Advanced Biology. In this video series on respiration in plants, we have already completed the glycolysis as well as pyruvic acid oxidation stages of the mechanism of respiration. Today we will start with the KREBS cycle. KREBS cycle is actually the third stage of mechanism of respiration. KREBS cycle is named after its discoverer Hans Krebs. It is also known as citric acid cycle, citric acid pathway, or KREBS cycle. Citric acid pathway because the first product that is formed is a citric acid. It is also known as tricarboxylic acid pathway. Why tricarboxylic acid pathway? Because there are many products that are formed in KREBS cycle are having 3 carboxylic groups in it, carboxylic acid groups in it. First of all, let us summarize the mechanism up till now. In the first stage, actually in mechanism of respiration, there are 4 main stages. The first stage was glycolysis. In glycolysis, the glucose, one molecule of glucose was converted into 2 molecules of pyruvic acid. This was a partial oxidation of glucose to pyruvic acid. In next stage, there was oxidative decarboxylation of pyruvic acid. Pyruvic acid was converted into acetyl coenzyme A. Acetyl coenzyme A. In this, decarboxylation was done. So, it is known as oxidative decarboxylation. Now, we will move towards the third stage, that is KREBS cycle. Glycolysis was done in cytosol of the cell, and then for aerobic respiration, pyruvic acid moves to the matrix of mitochondria. Pyruvic acid oxidation was completed in mitochondrial matrix, as well as this KREBS cycle will be done in mitochondrial matrix. Now let us start with the detailed explanation of KREBS cycle. KREBS cycle starts with the condensation of acetyl coenzyme A. This acetyl coenzyme A was formed in pyruvic acid oxidation. It condenses with oxaloacetic acid molecule, which is a 4-carbon compound. This acetyl coenzyme is a 2-carbon compound, and they both together forms citric acid. Hence this cycle is also known as citric acid cycle, because this is the first compound that is being formed. In this reaction, coenzyme A is removed and a molecule of water is being consumed. Now this reaction is being catalyzed by enzyme citrate synthase enzyme. This citric acid is 6-carbon compound, 2 carbons from acetyl coenzyme A and 4 carbons from oxaloacetic acid. Now in the next step, this citric acid gets converted into cis-aconitic acid. This is also a 6-carbon compound, and the enzyme that is helping it is aconitase enzyme. Molecule of water will be removed. In the next step, this cis-aconitic acid will get converted into isocitrate, again with the help of same enzyme. So in short, it can be said that citric acid is isomerized to isocitrate. This is also a 6-carbon compound. Now, with the help of isocitrate dehydrogenase, which enzyme? Isocitrate dehydrogenase enzyme. Dehydrogenase enzyme isocitrate will get converted into oxalosuccinic acid. Again, this is a 6-carbon compound. In this reaction, NAD will be reduced to NADH plus H plus. Now, in the next step, oxalosuccinic acid will get convert into alpha ketoglutaric acid. Alpha ketoglutaric acid. This is a decarboxylation process. Here a molecule of carbon dioxide will be released. So the enzyme is decarboxylase enzyme. Decarboxylase enzyme. Now as a molecule of carbon dioxide is removed, so alpha ketoglutaric acid will be 5 carbon compound. In the next step, alpha ketoglutaric acid will be converted into succinyl coenzyme A. So here coenzyme A molecule will be added. This is also decarboxylation reaction. So carbon dioxide will be removed. So this will become 4 carbon compound. Here again NAD is reduced to NADH plus H plus. So the enzyme used is dehydrogenase enzyme. Dehydrogenase enzyme. What is the function of dehydrogenase enzyme? Here also dehydrogenase enzyme was used. Dehydrogenase enzyme actually removes hydrogen atoms instead of oxygen in oxidoreduction reactions. Now in the next step, succinyl coenzyme A will be oxidized to succinic acid. Succinic acid is a 4 carbon compound. This was also a 4 carbon compound. In this reaction, the enzyme that is catalyzing this reaction is succinate thiokinase. Thiokinase. In this reaction, GTP will be converted into GTP. This reaction is known as substrate level phosphorylation. What do you mean by substrate level phosphorylation? Before that, this kinase enzyme is used where phosphorylation is to be done. Here, in this case, this is known as substrate level phosphorylation. Why? Because ATP is synthesized directly from the metabolite. The stage where ATP is directly synthesized from metabolites, that step is known as substrate level phosphorylation. This GTP will be converted into ATP soon. Here coenzyme A will be released out. Now in the next step, this succinic acid will get converted into fumaric acid or fumarate. In this reaction, the enzyme which is catalyzing this reaction is succinate dehydrogenase. All the enzymes of KREBS cycle are present in the mitochondrial matrix, but this is the enzyme which is present on the inner membrane of mitochondria. Now as dehydrogenase enzyme is there, so here FAD will be reduced to FADH2. In the next step, fumaric acid will get converted into malic acid. With the help of enzyme fumarase, fumaric acid is hydrated to malic acid. In the next step, this malic acid will get converted into oxaloacetic acid, or we can say oxaloacetic acid is being regenerated so that the cycle can be continued. In this reaction, the enzyme used is malate dehydrogenase. Malate dehydrogenase enzyme is being used. Along with that, NAD gets reduced into NADH2. So this is the KREBS cycle in which acetyl coenzyme A from pyruvic acid oxidation step condenses with the oxaloacetic acid, and after many steps oxaloacetic acid is being regenerated. In this cycle you have to memorize all the enzymes that are being used. These are the enzymes. In first step citrate synthase is being used. In next step aconitase enzyme used. After that, when isocitrate gets converted into oxalosuccinic acid, then the isocitrate dehydrogenase is used. Here decarboxylation is being done, so decarboxylase enzyme is used. Here again dehydrogenase enzyme is used, and then in next step succinate thiokinase enzyme is used, in which in this step very important thing to remember is this is a substrate level phosphorylation, and then succinate dehydrogenase enzyme converts succinic acid into fumaric acid, and then fumarase converts fumaric acid into malic acid. Then malic acid regenerates oxaloacetic acid with the help of malate dehydrogenase. So it's very important for everyone to memorize all the enzymes and understand the functions of particular enzymes. Now if we talk about the ATPs synthesized in this Krebs cycle or citric acid cycle, the number of ATPs synthesized is in this step. As I told you, GTP gets converted into ATP immediately after it is being formed. Also, in the second stage, two molecules of acetyl coenzyme A were formed. So, there are two GTPs used to form two GTPs. So, net gain of ATP is 2 ATP. Rest energy currencies, these NADH2 which are being formed, FADH2 formed. In the three stages NADH are formed, NADH2, and in one step FADH2 is formed. All these will be oxidized, reoxidized in the next stage, that is electron transport system, and then they will get converted into ATPs. I hope you might have understood the full process of Krebs cycle. If you have any doubt, you can write in the comment box. Your doubts will be clarified. Thank you.